So in this lecture we will discuss about the bacteriological culture media. Bacteriological culture media is basically a composition, a complex mixture of variety of different ingredients. Ingredients like peptone, yeast extract, meat extract, some carbohydrates, some sugars, sometimes the amino acids or some protein supplements which help in cultivation of bacterial cells is called as a culture medium. So culture medium is the medium which is nutritionally rich, which provides all necessary things to the bacterial cell growth, and it is used for the cultivation of bacterial cells. So these are some of the common ingredients which are present in almost all bacteriological culture media. The first ingredient is the peptone. Peptone, it is a complex mixture of partially digested proteins which are from the natural origin. It acts as a source of protease, amino acids, inorganic salts, that is phosphates, sulfates, some ions like potassium, calcium, and variety of different growth factors like vitamins, vitamin B complex. So peptone, it is a rich source of protease, amino acids, these are inorganic and organic salts, and the growth factors, that is the vitamins. That is why it is added in almost all bacteriological culture media. Yeast extract, the name itself reveals, it is the extract obtained by growing the yeast cells. So it is obtained from the baker's yeast, Saccharomyces cerevisiae. This yeast extract, it is obtained by growing the yeast cells, and after the growth the yeast cells are separated and filtered out. The extract, it acts as a rich source of carbohydrate, amino acids, and variety of different growth factors or vitamins. The third common ingredient which is present in the bacteriological culture medium is the meat extract. It is also called as beef extract. It is obtained from fresh lean meat or beef by hot water extraction. It acts as a source of peptone, protease, amino acids, creatine, creatinine, purines, minerals, carbohydrates, and variety of different growth factors. The meat extract, it is nutritionally rich as it provides almost all necessary things for the cultivation of bacterial cells. It is a rich source of peptone, it is a rich source of protease, variety of amino acids are present, variety of purines and bases are present. It provides different minerals like calcium, sulfur, phosphorus, and it is a rich source of growth factors as well as carbohydrates. Agar, that is agar powder, it is another important thing which is present in the bacteriological culture medium. Agar, it is obtained from the seaweed algae which belong to a category of agrophytes. So as the name agar, it is obtained from seaweed algae which belong to Agariaceae family, and that is why the name agar is given. So this agar, it is a pale yellowish compound which has a good gelling property, and that is why it is used as a gelling agent in the preparation of solid culture media. Agar, it dissolves in water, and in a concentration that is 1 to 2%, it forms a solid gel, tough gel, and that is why it is used for the preparation of agar plates or solid media over which the bacterial cells can be inoculated. Another important constituents of bacteriological culture medium is the water. Almost all media, they are prepared in water. Water, it is a universal solvent for all other things, and it is the main component of bacteriological culture medium, as the cells, any microbial cells, Bacteria is not an exception. They cannot grow without water. Water is essential, that moisture is essential for their growth, for their metabolic activity. Sometimes distilled water is used, sometimes tap water is used for preparation of the bacteriological culture media. But sometimes it is specifically given that copper distilled water must not be used for the preparation of bacteriological culture media. Copper distilled water, it contains copper ions, and these copper ions, they are bactericidal or bacteriostatic. The copper is lethal for the bacterial growth, and that is why sometimes it is specified on the dehydrated media bottles. Along with the asterisk, it was given on the instruction that avoid copper distilled water. Next, we have the classification of bacteriological culture medium. So, bacteriological culture medium, they can be classified based on the physical state as a solid media, liquid media, and semi-solid media. Solid media are called as agar media, liquid media are called as broth media, and semi-solid media, they are the soft agars or the broth with very little quantity of agar. Nutrient agar is an example of a solid medium which contains generally 1.5 to 2.5% of agar. Agar is used as a gelling agent and it is used in this concentration, that is 1.5 to 2.5% weight by volume. Liquid media, they do not contain any gelling agent, so no agar is present. Nutrient broth is an example of the medium. It is the same composition except that of the agar. It is nutrient broth. Semi-solid media, they contain very little quantity of agar, that is 0.2 to 0.5% of the agar. So the broth with very little quantity of agar, 0.5%, is an example of a semi-solid media. Depending on the oxygen requirement, the culture media can be classified into aerobic media and anaerobic media. The aerobic media is the media which is used for the cultivation of aerobic as well as facultative anaerobic cells. Example is McConkey's broth. McConkey's broth, it is used for the cultivation of E. coli. E. coli is an example of facultative anaerobic. So aerobic media, they mostly used for the cultivation of cells which require oxygen for their cultivation. Anaerobic media, so for those cells for which oxygen is deleterious, anaerobic media is preferred. So anaerobic media, it is made up of complex ingredients which help utilize the oxygen and avoid the oxidative damage to the bacterial cells. Anaerobic media are specifically designed for the anaerobic cells or stringent anaerobes, the cells whose growth can be inhibited by little concentration of oxygen. Example of anaerobic medium is Robertson's cooked meat medium. So Robertson's cooked meat medium, it is an example of anaerobic culture medium. Depending upon the chemical composition, the bacteriological culture media can be classified into simple or basal media, synthetic and defined media, non-synthetic, complex and undefined media. Now what is basal media? So these media, they are simple in composition and these media form the basis of all other media. So solid medium and liquid medium, nutrient agar and nutrient broth, they are the examples of solid and liquid media respectively. Peptone water is another example of liquid medium. All other media, they are basically prepared from the same composition by minor changes, by doing certain modifications in the basal medium. Synthetic media, the name itself reveals, it is derived from all the ingredients which are basically from the synthetic origin. As we know the exact composition of the medium is known as the defined medium. The formula is well defined. It contains pure chemicals, basically from the synthetic origin, complex, non-synthetic and undefined media. So here the formula is not known. The ingredients, they are basically from the complex origin. So the complex substances basically from the biological origin, such as milk, blood, egg, beef or yeast extract, these are included in the medium as a complex ingredient. And these media basically they don't have a fixed composition. That is why they are called as undefined medium. So example of this type of medium is blood agar. Blood agar is a complex ingredient which actually contains animal blood, sheep blood, synthetic and defined media. So here you can write the composition like citramide agar. Citramide agar is a synthetic medium. Nutrient agar is a basal medium. Depending upon the functional types, these are the variety of different types of bacteriological culture media. So bacteriological culture media, they are sometimes enriched media, enrichment media, selective media, differential media, transport media, assay media, and storage media. So from these names, you have got some sort of idea. So storage media, it is used for the long-term storage. Assay media, it is used to carry out variety of assays, microbiological assays. Transport media, it is used for transportation of delicate or sensitive microbial cells. Differential media and selective media, they are used for specific microorganisms for differentiation purpose. Selective media, they are used uh, for isolation purpose. Enrichment media and selective media, they are both same, but enrichment media, they don't have any agar, and that is why they remain in the liquid state. And this enrichment media, it is used for inhibition of unwanted cells. Enriched media, it is the medium which is basically enriched by adding complex ingredients from the natural origin, such as blood, milk, serum, yeast extract. So blood agar is an example of enriched media. So these media, enriched media, they are prepared for fastidious bacteria. Basal media is combined with blood, egg, serum, and we prepare the enriched media. Enrichment media, so when specific inhibitory substances which are added to the liquid medium which inhibits unwanted bacterial cells, the medium is called as the enrichment medium. Similar composition in the solid state is called as a selective medium, and these media, they permit only the growth of specific bacterial cells. That is why it is used for the isolation of specific bacterial varieties. For example, McConkey's agar, it is used for the isolation of E. coli. McConkey's agar contains inhibitory ingredients like crystal violet, which suppresses growth of gram-positive contaminants. Additionally, McConkey's agar contains bile salts or synthetic bile derivative, sodium torcolate, which inhibit the growth of non-enteric cells. Differential media, so these are used for differentiation. So this is used for the distinguishing between the different varieties of bacterial cells. Blood agar is used for differentiation of hemolytic cells from the non-hemolytic cells. McConkey's agar is used for the differentiation of lactose fermenting coliform bacteria from the non-lactose fermenting bacteria. Transport media, it is used for delicate bacterial cells, the cells like Neisseria gonorrhoeae. For these cells, a special medium is required which is known as Stewart's transport medium. So these cells, they are very sensitive. The cells are very sensitive to rough handling. To avoid any changes in the cells or to avoid any damage to the cells, they are transported across a specially designed medium which is called as the transport medium. Assay media, so as these media, they are used in the assays of different compounds such as antibiotics, amino acids and vitamins. The media, they have the special composition and the media is basically free of ingredients which can affect the taste results. And the last ingredient is the last medium is the storage medium. For long-term preservation of bacterial cells, for prolonged preservation, the storage media are preferred. Example is nutrient agar stabs. So nutrient agar stabs is an example of a storage media.